Hello students, welcome back to my channel CSIR Buddy, and the topic for today is glycolysis. This is the part 1 of this topic and the next part would be coming soon. So starting with the topic glycolysis. So first of all, what is glycolysis? So glycolysis is a process in which we break down a glucose molecule into further pyruvate molecule. So we can say glycolysis is the breakdown of glucose molecule. Now coming towards the steps involved in glycolysis. So these are the steps. First of all, glucose is a 6 molecule compound. This is a glucose molecule. Now take a plasma membrane of a cell. So glucose is water soluble, and our plasma membrane wouldn't accept the glucose molecule as it is. So for that we need a transporter. So that transporter is known as glut transporter. What do we mean by glut? Glut means glucose transporter. So now the glucose would enter the cell through this glucose transporter. But after entering this cell, the glucose may return to the cell, because this glucose molecule is this glut transporter is bidirectional. So glut transporter bidirectional है तो glucose को वापस भी ले जा सकता है. So in order to stabilize this, we add a phosphate group to this molecule. So a phosphate group is added to this molecule. So now this molecule is known as glucose 6-phosphate. And the enzymes which are involved in this are hexokinase. If we talk about muscle, so hexokinase is an enzyme which is primarily present in the muscles, and glucokinase is a type of enzyme which is primarily present in the liver. So the points to remember: this is present in the liver and this one is present primarily in the muscles. Now what's gonna happen? Now this glucose 6-phosphate would convert itself into a fructose 6-phosphate. So now glucose is converting this molecule into fructose 6-phosphate. And the enzyme which is involved over here, which will convert glucose 6-phosphate into fructose 6-phosphate, is known as phosphofructoisomerase. Why we are converting glucose 6-phosphate into fructose 6-phosphate? Because glucose 6-phosphate is a stable compound, is more stable than fructose 6-phosphate, and fructose 6-phosphate is somewhat unstable. So fructose 6-phosphate is easy to break. So therefore we convert this molecule into fructose 6-phosphate, and the enzyme involved in this is phosphofructoisomerase. Now what do we mean by isomerase? So isomerase enzyme, isomers are those molecules which are similar in their molecular formula. formula, but they only differ in their structure. So fructose, glucose 6-phosphate and fructose 6-phosphate are having the same molecular formula, but they have the different structure. Now again, fructose 6-phosphate would change itself into fructose 1,6-bisphosphate. So 1,6-bisphosphate, and the enzyme involved in this, we will call this enzyme as phosphofructokinase 1. And whenever we talk about kinase, so it is something related to adding a phosphate group. So this was carbon number 1, carbon 6. So 1 fructose was already added in the carbon number 6, and 1 we add in the carbon number 1st. So therefore the names 1,6-bisphosphate is given. Why bisphosphate and not biphosphate? Biphosphate नाम तभी मिलता है जब phosphate groups are very adjacent to each other. If one phosphate group is here and another is here, then it is known as biphosphate. If they are far apart, far away from each other, then it is known as bisphosphate and not biphosphate. From where we are getting this phosphate group? So we get this phosphate group via using an ATP molecule. When ATP molecule gets converted into ADP molecule, then we get this phosphate. So we are using one phosphate in this step and another phosphate in this step, wherein we are adding a second phosphate group. So ATP is converted into ADP plus phosphate. So these were the steps till now. I think this is clear to you all. As we can see that there is, in this step, it is only one directional. There is no going back from there. This one. Now going further. 1,6-phosphate now breaks down into 2 molecules. So this fructose 6-phosphate is breaking down into 2 molecule. और यहाँ से हम दूसरी स्लाइड में देखते हैं. तो fructose, what is gonna happen? Fructose 6-phosphate will break down into 2 molecules, and these molecules are named as dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Because carbon number 1 over here was consisting of a phosphate group, क्योंकि यही molecule हमारा break down हुआ है. 6 molecule compound था, 3 molecule compound में break down हो चुका है. So the number 1 carbon will consist of a phosphate group over here, and again number 3rd, in the number 3rd, second one phosphate group will be present. And the enzyme involved in this process is known as aldolase enzyme. Dihydroxyacetone phosphate get converted into glyceraldehyde phosphate 3-phosphate. Most of the dihydroxyacetone phosphate gets converted into glucose glyceraldehyde. I am writing this in a shortcut. Glyceraldehyde 3-phosphate. तो पहले dihydroxyacetone phosphate change होता है glyceraldehyde 3-phosphate में, then only the further steps go on, right? So the enzyme involved in this is known as triose phosphate isomerase. And what is isomers or isomerase, I have already mentioned. So now what's gonna happen is one enzyme will come which is known as glyceraldehyde 3 phosphate dehydrogenase. डीहाइड्रोजिनेज मतलब एक हाइड्रोजन आयन चाहिए, हाइड्रोजन आयन डिलीट हो रहा है. हाइड्रोजन आयन कहाँ से डिलीट हो रहा है? ग्लिसरल्डेहाइड 3 फॉस्फेट से एक हाइड्रोजन आयन ले लिया जाता है एंड NADH कन्वर्ट्स इनटू NADH वाया extracting a hydrogen ion from this molecule. As the name suggests that glyceraldehyde 3 phosphate dehydrogenase. So dehydrogenase का मतलब ये होता है. Then what will happen? So now this molecule which was glyceraldehyde 3 phosphate would convert into this one molecule with 2 phosphate groups. And this molecule, अब इस molecule को बोलेंगे 1,3 bis phosphoglycerate. Now this molecule will be known as 1,3 bis phosphoglycerate, right? So in the next step, one phosphate would be removed now, and when we remove one phosphate group, then we give this phosphate group to add this phosphate group into an ADP and convert this ADP into ATP. Now because this step is going twice. First, dehydroxyacetone phosphate is also converting into this step. So these molecules will be generated twice. So ATP, 2 ATP, ADP would be converting into 2 ATP. Phosphoglycerate kinase. And whenever we use this word kinase का मतलब होता है when we add a phosphate group to someone. So here we are adding a phosphate group to an ATP molecule and we are generating 2 ATPs from here. And now in the next step we are shifting this phosphate group into the second carbon. So this molecule is known as 2-phosphoglycerate and the enzyme involved in this is phosphoglycerate mutase. Phosphoglycerate mutase. So these were the steps. Now coming to the next step. Now in the last step we will be generating a pyruvate and will remove this phosphate group. So the enzyme involved in this process is known as pyruvate kinase. And because we are removing a phosphate group, so we are generating an ATP also, because this phosphate group will be passed onto one ATP molecule. So ADP molecule would be converting into 2 ATP molecule. And because whole this process is occurring 2 times, so there will be 2 of glyceraldehyde 3 phosphate, there will be 2 of 1,3 bis phosphoglycerate, and there will be 2 of 2 phosphoglycerate and 2 pyruvate. So in the end we will get 2 pyruvate molecules from this whole process. So an overview to this is, because glycolysis, glucose molecule is water soluble, and because it is water soluble, it becomes difficult for a glucose molecule to enter the cell, and for that we need a channel protein, and that channel protein is known as GLUT. Then in the first step we stabilize this glucose molecule via adding a phosphate group with the help of the enzymes hexokinase and glucokinase. Then in the next step we converted this molecule with the help of fructoisomerase into fructose 6 phosphate. देन आफ्टर दैट फॉस्फोफ्रक्टोकाइनेज की हेल्प से, काइनेज 1 की हेल्प से, वी एडेड ऑन अ फॉस्फेट ग्रुप टू कार्बन नंबर 1 over here and used an ATP molecule for the same. Then fructose 1,6 bis phosphate was converted into two molecules. These two molecules were dihydroxyacetone phosphate and glyceraldehyde 3 phosphate, and for this we used an enzyme that is known as aldolase. Then next enzyme was involved, glyceraldehyde 3 phosphate dehydrogenase, in which we dehydrated this compound. We took a hydrogen and gave to this molecule, that is NADH. Now following steps were done. We got 2 ATP and 2 ATP next. So total we got 4 ATPs from this reaction and 2 pyruvate, because this cycle is going 2 times. Now if we see a overview of this whole process. So 2 ATP तो हमने पहले यूज़ कर लिए थे. We got a gross of 4 ATPs in the last. So here 2 ATP, this one 2 ATP, 2 plus 2, 4 ATPs we got. But in these steps 2 ATPs were lost. So 2 ATPs were converted into 2 ATP. So if we count net ATP, then we will minus 2 ATPs which we have already used. So we will get a net of 2 ATP from this whole glycolysis process. And then in this step we got 2 NADP. So NADP कितना मिलेगा हमें? 2 NADP molecules, NADPH, and 2 pyruvate molecules. We got 2 pyruvate molecules overall from this process. So this was the net product which we got in the end of glycolysis. Now what is gonna happen to this pyruvate molecule? Either it will get converted into acetyl coenzyme A and will enter into the mitochondria, and at the last it will generate energy, that is ATP energy. But this step is possible only in the presence of an oxygen molecule. If there is no oxygen, no O2, then pyruvate enters a different pathway. So pyruvate enters a different pathway and gets converted into lactic acid. NADPH हमें कब काम आता है? जो ये NADPH हमें मिला था, NADPH हमारे तब काम आता जब हमारे पास O2 प्रेजेंट होता है और माइटोकॉन्ड्रिया में हमने यूज़ करा होता है NADPH को हमने यूज़ करा होता फॉर द जनरेटिंग ऑफ ATP एनर्जी. तो ये अपना हाइड्रेट यहाँ पर दे सकता था. बट नाउ NADPH हैव नो वेयर टू गो. तो NADPH गेट रिड्यूस्ड. सो NADPH gives its hydride to pyruvate and itself it get reduced into NADP. So and pyruvate get oxidized. So reduction होता है NADPH का और ऑक्सीडेशन होता है लैक्टिक एसिड का. Now lactic acid could travel further into liver, or it can lower the pH because it is acidic. Obviously this is gonna lower the pH. So pyruvate के पास दो जगह है जाने के लिए. या तो वो लैक्टिक एसिड में कन्वर्ट हो सकता है या तो it will enter the Krebs cycle and will be involved in generating more ATPs. So that was it for today's video. Thanks for watching my video. If you like this video, please like, share and subscribe to our channel and press the bell icon. Thank you so much.